I'm Stephen Jones. I'm head of bioinformatics and co-director at the Genome Sciences Center at BC Cancer in Vancouver. Most of the work that I do is involved in taking a DNA sequence of DNA sequencing machines and trying to interpret what that sequence means, whether it's involved in human disease or whether it's understanding our natural biodiversity, understanding whether there's a mutation there that someone might need to know about. And a large part of that, as you might think from the institution I come from, which is BC Cancer, is we're really pointing genomics in a large way at the disease of cancer. Cancer being, of course, a disease of the genome. It's the genome that becomes mutated and changes in such a way that the cells rapidly divide in an uncontrolled way. And a large part of what we do is to try and decipher what those changes are, help us understand what makes a cancer cell, what are the different ways that a cancer cell can form, and then importantly, do those mutations indicate any clues to us in terms of what the best therapy might be for that person. So it's a fairly broad mandate that we have, but everything we do basically involves the sequencing of DNA.
That's amazing. Thank you for that overview. So can you tell us a little bit about something new or surprising that you've learned lately in your work?
Well, one of the kind of more exciting newer projects that we're doing is working with the Children's Hospital here in Vancouver. And there's been an ongoing need, obviously, for many years of having children in the neonatal intensive care unit, where they do have an issue, they are not thriving, or they're actually very sick, but it's not clear what the diagnosis is, and typically there is a genetic reason or issue that, if we can understand more quickly, would allow for probably more targeted and more correct therapy for that child. So with that goal in mind, how do we sequence a very sick child in the neonatal intensive care unit as quickly as we can? We've started a program using a new type of sequencing called nanopore sequencing, where we can take a very small sample of blood from the child in the morning, and we can be generating DNA sequence from that child the very same day. Historically, or how it's done now, the blood is shipped off. So we're generating, or can be generating, sequence now while the blood sample is still in a FedEx package waiting at the airport. So now, of course, it's one thing to be generating sequence, but it takes maybe a couple of days or more to try and work out or to sequence the genome of that child fully, and also to do the relevant bioinformatic analysis to see can we tell what disease this child may have. So it's an ambitious project. We're excited because it clearly meets a very important need within the hospital environment, and it's very, very new technology, so we're learning to use that as well. But hopefully that'll improve the lives of some children in British Columbia.
Great. So what is something that you think other genomics researchers or users of genomics should know about in their work?
Well, I mean, that's a big question as well, but it is a very fast moving field, and I think one of the things that we've been looking at in much more detail over the last couple of years is how we can now not just sequence the 4 bases of DNA, but to understand that those bases can also have further modifications, and so this area of of study where we have these modifications in our genome. They're not typically inherited changes, but they're kind of bookmarks that our cells put on the DNA. And this area is called epigenomics, and it really mostly involves the addition of a methyl group to one of the bases. But this can actually be very important in understanding what's happening in that cell, what's happening in disease, how genes are being turned off or turned on. And what's interesting now is that we can detect those changes at the same time as we're doing a normal genomic sequence. So that, I think, opens up a bit of a larger world for our understanding of how cells work. And the fact that we can do it now with no extra cost at some level means that maybe other researchers would have the ability to ask questions or do experiments that they wouldn't otherwise previously have been able to do or conceivably afford. So we're very excited about how that works. The quality of the data is equal, or actually in some ways better than the previous ways that we were doing it, which involved extra experiments and obviously extra expense. So that's something that we're working very hard to get into people's hands.
That's very exciting. Are you able to maybe provide a specific example of how you're using it, or even like what the next steps are now that you have this technology and the ability to do this? What are some of the areas that you're going to be focused on with this?
So one of the areas that again, we've been working with researchers at the Children's and Women's Hospital, where again, we can have children with an undiagnosed disease, not necessarily in the intensive care unit, but having a failure to thrive or a developmental disorder of some kind. And so often we generate a genome, and we can find a mutation in a gene which is associated with the kind of disease the child has, with the phenotype of the disease. But it's a mutation we've never seen before. So we don't really know if that mutation is disease causing. But now with the methylation, what we also know is that children with that particular disease, for instance, have a very distinct methylation pattern in their blood cells as well. And so we can now look at the methylation pattern for those children and say, yes, actually, not only do they have a mutation in the gene, but the epigenetic pattern in their blood cells matches someone who has the disease as well. So now we can have a much more robust diagnosis for that child, and we can make a pretty strong assertion now that mutation is disease causing. We can put that mutation now in public data banks. So if any other child now is seen with the same mutation and the same kind of clinical features, that the clinician can be more assured that they're giving a correct diagnosis. So again, we're really excited about how that could speed up diagnosis. And certainly a lot of the clinicians we're working with at the hospital are very excited about the ability to do this.
Yeah, that is amazing. And that definitely sounds like precision medicine. So I think that is very exciting to hear that that's happening in real time now with the work that you're doing. So thank you for sharing that. Is there anything else in terms of the future looking ahead? Where do you think we're going maybe within this field in terms of the pediatric care that you're providing or other populations, or what else is exciting to you in terms of the application of genomics and healthcare?
Yeah, well, I mean, one thing, one advance that's been made in the last year, and I think this is a very salient and key advance, is the ability by a piece of software developed by engineers at Google called AlphaFold to actually take the sequence of a gene, translate it into what the protein would be, And then what AlphaGene can do is actually predict the three-dimensional structure of that protein, right? That to essentially go, if proteins are, the machine parts of our cells, to now actually have the three-dimensional structure of that computationally is phenomenal. That really is a game-changing moment in conceivably how we might design or think about experiments in the future. We have a biodiversity project where we're actually going out and looking at how we can sequence more genomes, get more genomic resources for the flora and fauna that live in Canada in particular. But what this does now is that we can, and we didn't think we could do this when we conceived of the program, was we can now sequence an animal within a couple of weeks. We've got some pretty good tools to computationally predict where the genes are. And now, once we've got the genes, we can have a pretty good prediction of the three-dimensional structure of every protein in that organism just in a few weeks. And that opens up all kinds of possibilities about how it might be potentially negatively affected more than other animals, for instance, by insecticides or pesticides, within the environment. So that really is a fundamental change in how I think we can do biology. It really has been a holy grail for 30, 40 years to can a computer predict the three-dimensional structure, and so now that's been done.
Wow, that is amazing. Thank you for sharing that. I haven't heard about that with Google, so that's very, very exciting. And I think it's very timely, considering that the theme for this year's World Health Day is our planet and our health, and how there is the linkage between the environment and our health. And there's obviously an overlap in the work that you're doing related to people's health and also the environment. And is there anything else that you'd like to share, maybe in relation to this, why the focus on biodiversity and in relation to climate change is so important? And it's amazing that we're doing this work in Canada. So is there anything else you want to share about that?
Well, yes. I mean, what CGEn has been able to do is to set up, sequencing infrastructure across the country. Most of the demand and the necessity has been obviously with human health, and so we can sequence now human genomes at scale, and at a population type basis. But what's nice about DNA sequencing is that all other plants and animals have exactly the same molecule. So we can point these sequencing machines and the downstream processing, the computational processing, at other organisms as well. And so one of the things that we wanted to do is not just be involved in human health, but how can we point CGEn's technology and expertise for all of the life within Canada. And so what we've been embarking upon, and it's a program funded by Genome Canada, is to work with an international consortium called the Earth Biogenome Project to start to get the genomes from a large portion of the plants and animals across the planet. And so we'll be focusing on Canadian plants and animals. And it's really going to allow us to develop tools where we can look at the genetic health of populations, that we can see how populations are changing. We can see if a population is struggling from its population size, and if it gets too small, then you have problems with the genetics of how that population can be viable in an ongoing way. So understanding that would allow us to make conservation changes maybe sooner than they would otherwise happen. Canada is the second largest country in the world. It has a huge interface with the Arctic. That's a place of very concerning environmental change due to global warming. And so that's, again, a place where we would like to be understanding the animals and plants that live in that area. And so our contribution, is going to be able to find what the genetics of those animals are.
Amazing. It's very exciting work and very important. So thank you for sharing that. Is there anything else that you'd like to add before we wrap up?
No, I don't think I've got anything else to.
Okay. Well, thank you again for taking the time to share all this really important work that you're conducting with your team. And we'll hopefully hear from you again soon. So thank you for taking the time.
Great. Thank you very much.